We'll get started for today. Thank you, everybody, for coming out in the wind and rain, for those of you who are here today. And welcome to the Conservation Lecture Series. Today's lecture is part of a series of lectures, and we have a website that you can visit if you would like to sign up for future lectures. And we also have videos and PowerPoints from past presentations if you'd like to check those out. You can contact me if you have any questions about the lecture series or about any of our presenters. Just to give you a quick look at what we have coming up in the lecture series, we have one next month on rare plants in Pine Hill. We're going to be having 2 lectures in February. One of those is going to be out of our Ontario office, so those of us in Sacramento will get to WebEx for that one. We'll have a talk on invasive water snakes in March and a talk on white nose syndrome in bats in April. And that one is in red because it's at a slightly different time than we normally hold the lectures at. So today, we're really lucky to have Dr. Sarah Kupferberg. She's going to be speaking about foothill yellow-legged frogs. And Dr. Kupferberg grew up in the East Coast in New York and spent her summers on the Finger Lakes region, which is where her love of aquatic ecology started. She went to Duke for her undergraduate degree as a botany major and moved to California after college. She worked doing environmental education for about 5 years and then went to graduate school at UC Berkeley. She worked in Mary Power's lab with a focus on algal-based food webs in rivers. And her current interest, or her initial interest in foothill yellow-legged frogs was studying tadpoles as grazers, and from there things have grown. So thank you very much for being here today.
Well, thank you very much. I really appreciate the opportunity to share with you what I've learned about foothill yellow-legged frogs over the last 20 or so years. I sort of stumbled upon them in my dissertation research, and there's, to me anyway, a lifetime of knowledge to be gained by studying them, and so I've just kept digging deeper, and I'm going to try to condense everything that I've learned about them since I started studying them into talk. So, please feel free to interrupt me if anything that I say doesn't make sense or print is too small on a slide and you can't read it. And I guess for those of you out there in WebEx land, you can type in a question and it'll be read to me. So the title of my talk today is From Algal Food Web Ecology to Dam Management, Connecting the Dots One Tadpole at a Time. And my focus is to try to cross different spatial scales and different temporal scales to connect what might be very small interactions and scale them up to the whole region that the species occurs in. And similarly, to take stressors that we're putting on the species at a very large spatial scale and try to bring it back down to the natural history of the species. There's lots of people to thank. First, I'd like to thank Scott McBain, who I work with, and he's an engineer who helped develop the model that I'll be talking about in the second half of the talk. And I've worked with people from academia, from different resource agencies, and some of this research has been funded by the San Francisco Public Utilities Commission, and before that, the California Energy Commission, and then before that, different independent grants when I was a student. So in California, we get more than 15% of our electricity from hydropower. As a state, we have more than double the number of dams of any other state. And we also have the highest biodiversity of any state in the U.S. And because of our Mediterranean climate, like on a day like today, we're getting our supply of water. It arrives in the winter, but most of our demand is in the summer when we have higher demand for electricity, higher demand for irrigation water, for drinking water. And as a result, there's an inherent mismatch and an inherent conflict. when we get our water and when we need it. There we go. So there are these inherent water, power, and environment conflicts. And across our state, there are many large projects that are being relicensed. And we have competing demands for water. And there's different regulatory agencies involved. Lately, I've been involved a lot with Federal Energy Regulatory Commission licenses for hydropower dams. But, with the passage of our recent Proposition 1, I guess, we're going to be having more dams built, and these conflicts are going to be ongoing. And there are many different players. There are the utilities, there's the municipal water districts, agricultural users, recreational boaters, people who go fishing in our rivers, commercial and native salmon fishers are interested in restoring habitat and access to streams for salmonids. And then there's the rest of the wildlife that depends on the water in the rivers. And because most of these projects are going to have operating licenses that go for a long period of time, there's a unique opportunity right now where we can have a long-term impact and have science-informed management that's really going to make a difference over a long period of time. So an example of the kinds of conflict that I'm talking about, I'm sure that you're all familiar with, is that dams block fish migration. So this is a picture of the eel watershed. It really only has one big large dam on it, the Scott Dam. That's shown in the box in the lower right corner. So for most of it, fish can swim up the main stems. So the red colors, orange, those are the larger rivers, and they're trying to get through those large rivers, which can get awfully warm in the summer, and get to the dark, cool, shaded tributaries for their juveniles to rear. Frogs, on the other hand, are moving in the opposite direction. The adults of the foothill yellow-legged frog often live in these steeper, darker tributaries, but because tadpoles eat algae, they need to be in a really warm, sunny environment where there's lots for them to eat. So the females will hop down out of their tributaries, and they go to breeding sites called leks, where many males congregate and set up territories, and there's even male-male aggression where they set up little calling territories. They mate, they lay their eggs, and then they go back to their home territory. And radio tracking has shown that I think the longest distance movement for a female was 7 kilometers in Tehama County. So that's but most of them don't move quite that far. But the conflict arises in terms of frogs versus fish when the migration is blocked for fish, and we're now expecting the fish and the frogs to use the same exact real estate in the river. And even though these species have co-evolved, they didn't always share the exact same inch or square inch of the river systems. They kind of were passing in the spring in different directions. So one of the biggest stressors of dams that they place on the system is by changing the flow regime. And there are many different kinds of altered flow regimes. What I've got plotted here are flows for several different rivers, some regulated, some not. And I'll be referring to these rivers later on during my talk. So on the x-axis is simply date for a representative year, 2005. On the y-axis is discharge. So that's the volume of water moving down the stream in any given second. And what you can see is for some regulated rivers, like on the Feather River, that you can either have a flat line hydrograph, that's the dashed line, dashed and long dash with a dot, that's below Poe Dam, and then below Cresta Dam, when the dam was first being relicensed, there was a period of time when they were experimenting with doing monthly pulsed flows for boating. So that's what's circled there, is boating, flat line, then another boating flow. Then you can have spill in the spring where the dam is full, snow is melting, and you get a big spike, high flow release, and then the water gets below the spillway, and you have a very rapid cessation of spill. Then you get snow melt again, it goes way back up, and then cessation of spill again. Or the most extreme kind of a unnatural hydrograph is when you have daily power peaking. So this is from the the Middle Fork American below the Oxbow powerhouse. And there, every day, to meet peak demands for electricity in the summer, they release water and then they turn the water off. And part of the timing also is to provide boating opportunities. And those all stand in contrast to what the unregulated hydrographs look like. So I've included the South Fork Eel River and Alameda Creek, and these are both much smaller rivers, smaller streams. And as you can see, the general pattern is that you have very gently declining flows over the course of the spring and summer. And that's the biologically active season when most of algal food web ecology happens. So the basic issue is that there's a mismatch between these impaired conditions and the adaptations of foothill yellow-legged frogs. On the left are the aquatic life stages that are very vulnerable to flow fluctuation. So there's a pair of frogs there in amplexus, and the females, you can't really see her because the male's on top holding on to her. And the males, if you ever are in the field and you want to tell a male frog from a female frog, in the spring especially, the males get what are called nuptial pads. So on their thumbs, they get really dark, rough tissue, and then it allows them, when they're in amplexus with a female, to hold on to her really, really tight. And then she goes around and looks for a good rock to attach her eggs to. And these frogs only occur in flowing water. So they have to find out-of-the-way, lower velocity places to attach their eggs to the rocks. And the females actually will scrape the rocks clean with her back feet. And she might go to one rock, try to scrape a little bit, it's not clean enough, go to another rock, and eventually she'll oviposit. And then usually over a week or two, depending on temperature, the eggs will hatch. But what's shown here are stranded clutches. So in a case where when a reservoir is being managed to fill and spill, and you get that very rapid cessation of spill, clutches can be left high and dry. And so that's what those little black patches are with the red arrows pointed. But luckily, you can still count them. So a lot of the census techniques rely on the fact that the females lay this one discrete clutch of eggs each year. That way, you can go out and count and mark the clutches of eggs, and you know exactly how large the breeding female population size is. It's pretty convenient. So assuming that there aren't any pulsed flows that have high velocity that can detach the eggs and they're not stranded, in about 2 weeks, the eggs hatch, and over the course of the summer, the tadpoles grow, and then by about the end of 3 or 4 months, the tadpoles start to undergo metamorphosis. And in some ways, this life stage is the most susceptible to flow fluctuation because they're not really great swimmers yet, and they're also have a pretty big body size. So the smaller tadpoles, if flows come up, they're small, they can get into the interstitial spaces, but when they're big and they've got rear legs that create drag, they can't swim very well, they can't fit into the interstices. So in swimming studies that I've done, actually the metamorphs are the most vulnerable to pulse flows and being washed downstream and maybe then into the mouths of a predatory fish in a pool. And then it takes them a couple of years as a juvenile until they are old enough to reproduce. So in Northern California, the life cycle is at least 3 years for a female to then become gravid, and gravid just means she's full of eggs. That's why that frog looks so fat. In Central California, the frogs can reach reproductive maturity in 2 years. And a lot of people ask, well, how long do they live? And most frogs probably don't live as long as this frog. And this is thanks to my friends at Garcia and Associates who have been studying the frog population on the North Fork Feather River for Pacific Gas and Electric. And they started taking chin photos of all the frogs that they caught, and they also did pit tagging to verify. But the frogs have unique spot patterns on their chins. So this female was first captured in 2004, and she was already a very large female that had to have been at least 3 or 4 years old based on her size. And she's been recaptured all of these times through 2012. I don't know. She may. I'm not sure if she was captured this last season or not. So they can live pretty long, but I would imagine that most females breed once or twice, and that's it. But there are some of these really large, sort of, grand dame frogs out there that are pretty amazing. So the effects of hydrologic alteration has impacts across scales. At a large scale, across the West, we're reliant on hydropower, so we have regional issues, and then within a watershed, there are also impacts, and then the effects are also very small on, the tiniest individuals of eggs and embryos and small tadpoles. And across this spatial scale, it also parallels variation in time. So across a broad perspective, we can look at range-wide changes that occur over a period of 25 or 50 years, and I'm going to be making comparisons between the historic and the modern status of the species, and then also make comparisons of what the average density is of populations that are in regulated versus unregulated rivers. Then I'm going to look at individual population trajectories over a time scale of 5 to 20 years, doing time series analyses, and then I'll be looking at much shorter term effects over minutes, like whether or not an egg gets scoured or a tadpole gets swept downstream, over months and years. So range-wide changes over the last 50 years were studied by my friend Amy Lind, and what's shown here is a distribution map of the species range. That's the yellow blobby part of the picture. And these frogs are endemic to California and southern Oregon. They don't occur anywhere else. And each of those little blue triangles is a dam. So you can see there's this huge overlap between dams and their range. And what Amy did is she looked at historic locations. She used all different museum records and CNDD points, and then she randomly chose 10 points from every county, 10 localities from every county across the range. And then she went back and either visited those sites or got in touch with local experts to say, is this population still there or not? And she then looked at whether or not there was a dam upstream of those sites, how big was the dam, how far away was it, how many dams were there. And what she found is that the frogs were absent from more than half of their historic localities. And the absent localities had a significantly higher number of dams upstream, and also the absent localities, when there was a dam, it was significantly taller. So the bigger the dam, the more water it holds behind the dam, and then hence a greater change to the flow regime. So that's a longer time scale. What about of the populations that have survived? So over half are gone, and among the surviving ones, we went through all the different FERC relicensing studies, academic studies, and we found 27 populations that had fairly good to egg mass censuses, where people went out repeatedly during the spring, captured the whole breeding season, so they went out time after time and could count how many egg masses there were. So we compared the average number of clutches per river kilometer. And it's a little hard to compare small rivers, big rivers, and that's why we've done standardizing it by the length of the river. And what you can see is the black dots are regulated river sites, and the white dots are the unregulated. And there's like 5 to 6 times more frogs in a typical unregulated river than those in the regulated. And amongst all of these sites, there were 6 watersheds that, along with my colleague Alessandro Catanazzi, where we also monitored water temperatures, and we had paired rivers, regulated and unregulated, within the 6 watersheds that are up here. And for 3 of these watersheds, there's actually very good time series data. So we're getting into shorter and shorter time scales now. So the population trends can be related to lots of different stressors. The first one I'm going to talk about is the South Fork Eel River, which is unregulated, and it's a fairly healthy, dense population. You have to notice that there's a different scale on the y-axis of each of these 3 graphs in terms of the number of clutches per kilometer. And the first thing you can see is that there are periods of boom and bust, and that's natural. We have flood years, we have drought years, but generally, because these frogs are really fecund, each female can lay 2,000 eggs. In one good year, the population can rebound, and then slowly it declines over time until there's another bonanza year. And this is a fairly typical kind of population dynamic for frogs. So when you do a long-term time series analysis, even though if you look at any 5-year time period, it might look like it's vastly going up or vastly going down. Over the 20 years, there's actually no significant trend through time. So then the next graph is the North Fork Feather River. And you'll remember from the hydrographs that I showed you that the Cresta Dam is where they tried doing these pulsed boating flows. And initially, for the first 3 years of when they did the boating flows, the frog numbers stayed constant, even though the pulses were causing a lot of mortality by scouring. And what happened was it took that 3-year lag time, so when there was no production of cohorts, the effect didn't start to show up for 3 years. So you'll see that the 2 rivers are going sort of in parallel for the first few years, and then they diverged. And even after the divergence, then everybody figured out, hey, this is not a great idea to have pulse flows during the breeding season, and so that was changed. But unfortunately, the frogs really have not recovered since then. Part of it has to do with simultaneously with ending the boating flows, there's overall higher flows, and so there's less inundation of the proper habitat, so their breeding habitat is now higher up. And so it's not exactly clear what it's going to take or how long it might be for the Cresta population to rebound. And that really ancient female that I showed you is from Cresta. So right now it seems like that population is being maintained by these really old females, and they need some serious recruitment to recover. And then the last time series is for Alameda Creek, and in the unregulated reach, it's different from these other rivers in that it's an ephemeral stream very high up in the watershed, and so as you can see, the numbers have been declining over the drought in the last few years, and in the regulated reach, which normally is much lower than the unregulated, in this last year, that's where all the permanent water was that leaks out of the dam, actually is keeping the channel wet. So in a sort of odd turn of events, it looks like, in this case, having a dam that maintains a more permanent flow has actually made it so that for the first time in this very long time series, there were actually more frogs breeding downstream than there were upstream. And also in Alameda Creek, it's complicated by other stressors, like bullfrogs are present in the system, and that's the really big tadpole there in my hand. Another stressor has to do, in addition to changing the flow regime, has to do with changes in water temperature downstream of dams. So this is O'Shaughnessy Dam on the Tuolumne River, and in the spring, when Hetch Hetchy is full, water can go over the spillway, and that's taking water off the surface of the reservoir. But as summer progresses and the reservoir elevation declines, water can't flow over the top from the spillway, and the water gets released by valves at the bottom of the dam. And that water coming out in the middle of the summer is 12 degrees C. So it takes many kilometers for that water to warm up as it moves downstream. So what I have plotted here is the realized thermal niche for the frogs of where they can actually breed. So in our 6 different watersheds where we put out thermistors along a longitudinal profile in the river, so we'd be right below the dam and then going downstream and then try to match that same elevational range in an unregulated river in the same watershed. So on the left, I have plotted the Sierran sites, and the right are our coast and cascade sites. And what you can see is that the dark dots are the populations that are more dense, where we found at least 5 or more clutches per kilometer. And that's still pretty low. Like if you remember the South Fork eel, like in some years it was in the hundreds per kilometer. So this is sort of a low bar for saying what's a dense population versus sparse. So that was our cutoff, less than 5 is sparse. And if there's red around the symbol, it's from an unregulated river, meaning it's warmer. And the blue are for the regulated. And you can see that generally it's a lot colder in these regulated sites, and the populations are also smaller. So in the Sierra, we have a much wider elevational range. In the coast range, it's a much narrower elevation range. And interestingly, the frogs in the Sierra seem to be occupying a much warmer set of temperatures than on the coast. And I think part of that is because overall the growing season is shorter, so the tadpoles, their development rate is controlled by temperature. So the shorter the growing season, the warmer they actually need it to be. So now I've covered sort of the large spatial scale and the longer time scale patterns at the scale of decades, and looking at populations and range-wide status and the conditions that they face. And now I'm going to focus much more on the small-scale things that are near and dear to my heart, because I really just love doing experiments and manipulating conditions and moving tadpoles around. I'm going to present the results of some rearing experiments, looking at the thermal performance of tadpoles, how their growth responds to the food quality of the algae. So in our Mediterranean rivers, there's an algal succession that occurs as the flows recede in spring. So in March, there's just sort of a layer of fine sediment and not much is growing. And then the algae starts to regrow from little basal pads where it's attached to the rocks. And one of the dominant species of algae in California rivers is called Cladophora glomerata, and it's a filamentous green, and it's called Cladophora, like I'm sure you've all seen cladograms looking like a tree, and it's called Cladophora because the filaments branch. And as a result of that branching, which you see in the center, it creates an incredible surface area for single-celled diatoms to attach to. Those diatoms will overgrow the algae, and then that's really what fuels the food web. And it gets consumed so that by August, these big, long turfs of algae are now mostly been gobbled up, and like here, it has like a webbed appearance, and that's from little midges that weave tufts out of the algae, and then they actually graze the epiphytes. So they make a little home for themselves, and they go off, and they just go outside their little tuft, eat the diatoms, and go back inside. And this is what it looks like microscopically when the filaments, instead of being bright green, they'll start to look rusty red and kind of, you probably think, ooh, gross. But it's actually, that rusty red color is from the diatoms that grow as epiphytes. And there's one group of diatoms that appears late in the succession, and they will proliferate when nitrogen becomes limiting in the water as the summer progresses. So the green algae is sucking up all the nitrogen. And then these species that have cyanobacterial endosymbionts have an advantage because the endosymbionts can fix the gaseous nitrogen that's dissolved in the water, and they have a competitive advantage. And they can completely cover the surface, and that's what you see in the scanning electron micrograph, is that the surface of the algae is completely covered with diatoms. Foothill yellow-legged frog tadpoles are incredibly efficient at scraping all of those diatoms off of the algae. So I've done experiments where I take a cobble that's covered with algae and it looks totally rusty red, and I'll enclose it with tadpoles, and if I put in tree frog tadpoles, it continues to look rusty red. If I put in Rana boylii tadpoles, within a week, the turfs on those rocks turn bright green again because they scrape all of the algae off, and then they recycle the nutrients by pooping on top of it. And it's because they have this very unusual mouth part where they've got rows and rows of keratinized teeth. So what you see is the beak of the tadpole, and then these rows of scraping teeth that if you looked at those under a scanning electron micrograph, they actually look like a rake. So they go along the filament of the algae, and they're essentially like scraping and raking all of the diatoms off. So they're very important in aquatic food webs. They're converting algae into snakes or into dragonfly nymphs or to all the different things that eat tadpoles. Because if you think about it, if every tadpole turned into a frog, we would be up to our eyeballs in frog because there are thousands in every clutch of eggs. So they're a really key element in algal-based food webs. So I reared tadpoles in different temperature streams. So at the Angelo Reserve, which is part of the UC Natural Reserve System, there's a number of different creeks, some that are narrow, shady, cool, others that are much more wide open and sunny. So I stocked laundry baskets that I glued mesh over with tadpoles and rocks covered with algae, and monitored the temperature in all of my enclosures, weighed the tadpoles every week, saw how long it took them to reach metamorphosis, how many of them survived. And in some cases, I made them eat the algae that grew in that stream. So if it was dark, there wasn't a lot of algae. In other enclosures, I supplemented. I took the food that grew in a warm, sunny place, but made them just eat that food in a cold place. And what I found was that there was this interesting interaction between water temperature and food quality. So at really low temperatures, where it's only 16 degrees on average in July, the ones that had to eat the ambient periphyton, none of them survived to metamorphosis. But if they had supplemented food, they could survive. And that difference kept narrowing as the temperature increased. So at the warmer temperatures, it didn't matter if they were getting the best food or not, right? Because the diatoms, they're full of protein because they have the nitrogen fixers. And also I forgot to tell you that unlike terrestrial plants, when they make excess photosynthesis, they make it into starch. So think about potatoes, right? Potato plants doing photosynthesis, it's taking that food and storing it in its roots as starch. Well, diatoms don't do that. They store all of their excess productivity as lipids. So it's the diatoms are both high in protein and high in fats. So they're kind of tadpole superfood. So what's interesting is that the thermal preference of tadpoles matches their growth performance. And we put tadpoles into long gutters where we heated one end, cooled another end, and we saw what was their temperature range. So basically, they chose between 16.5 and 22. And that's also when they grew the best. And what I've plotted here are comparisons between a river, the main stem eel that's getting hypolimnetic releases from Scott Dam versus the unregulated South Fork eel. So the South Fork eel, you can see by midsummer in July and August, when tadpoles are growing really fast, it's in the preferred range. Whereas in a regulated river, it doesn't warm until much later in the season. And when we went all over the eel watershed and saw how dense are the populations, and we looked at how quickly does the water warm, what's the mean temperature, and we came up with this composite measure of temperature in a principal components analysis, and so that's on the left. And as you can see, there's a real spike in terms of the density of frogs matches the temperature. So on the right, I've plotted it just as temperature so you can see where the spike would be. So right where the middle of their preference range, where they grow the best, that's also where the populations are most dense, which is actually pretty amazing that the conditions that suit larvae the best seems to be determining the distribution of the population in general. So it really seems like that recruitment is super important to their populations. So I know a lot of you are really interested in looking at habitat areas. You write habitat conservation plans, and certainly habitat is really important of having a place, having that piece of real estate. But I'm trying to look more mechanistically about how to determine what's going to cause success or failure of a cohort of tadpoles. And instead of just looking at stream plotting on the x-axis, stream flow, and how much usable area there is based on different levels of discharge, I wanted to say, what's the whole suite of conditions? And with Scott McBain's help, we developed a model to look at how to use the fluctuations in stream flow, how to look at temperature changes to make it have a more mechanistic understanding than just say, let's wet this amount of rock and assume we're going to produce more frogs. So what our model is a very simple Excel spreadsheet model, and it's based on a particular cross-section of the channel. And it works at a daily time step, going through a bunch of yes-no questions and saying, well, are they still alive or are they not alive? Going through the daily time step as we vary flow and as we vary temperature. The window is it starts at breeding, it ends at overwintering, and we use a multi-year time series of daily data. So we go through year by year by year using historic flow data and sometimes either historic temperature data or modeled temperature data and say what would be the success under each of these years. And we assess the fate of the eggs and the tadpoles each year. And then we can predict the changes in reproduction success as a function of the discharge, water temperature, the shape of the channel, how deeply the frogs are laying the eggs, breeding dates, and other parameters. So the computational process is based mostly on the immobile life stages. So we have these theoretical clutches of eggs that are laid, and we can set what depth different rivers, frogs lay their eggs at different depths. So we can put these hypothetical egg masses, that's what those little blue spiky balls are. So we put them out into a specific cross-section that we've measured in the river. And we know what the relationship is between discharge and the stage, and discharge and velocity. And we can say, okay, this egg is laid on day 1. Now we know what the temperature is going to be. That egg has to stay wet, maybe for only 10 days if it's warm. It might have to stay wet for 21 days if it's cold. So by having this daily time step of temperature and discharge, each day we can say, is that clutch still alive or not? And we can also say, has it been scoured away based on an empirical relationship between flow fluctuation and what it takes to scour an egg mass downstream. So what happens in regulated rivers, in addition to changing the temperature and the flow, the morphology, the shape of the channel also changes. And what I've shown here is a little cartoon of two different types of channels and a frog laying her egg at the water's edge. And so the frog is not to scale. She's huge. But in an unregulated river, you generally have a much more complex type of channel. And as a result, in a wide, shallow channel with lots of complexity of bars and not a lot of vegetation encroaching, there's going to be a less steep relationship between changes in discharge and either changes in velocity or changes in stage, right? So just think about if you've got like think about two different shapes of champagne glass, right? So you can have the tulip-shaped champagne glass, or you can have the ones like this, and you're pouring water into it. So you're pouring equal volumes. Which one is the height going to change more quickly as you pour in? The tulip one, right? So downstream of a dam, you often get encroachment of vegetation along the sides, and you get channel incision in the middle, and the channel starts to take on that much more U shape, as opposed to a big, wide, flat shape. And as a result, then as you put more water down, velocity is going to speed up along the margins, so that puts an egg at greater risk of being scoured away. And if flows drop, it puts an egg at much greater risk of being desiccated, right? Because the stage is going to drop faster. So we applied this model to three different systems: the Trinity River in Northern California in Alameda Creek, and then also on the Tuolumne River. So first, the model assesses if the days of inundation are greater than the time needed for embryos to develop. And that's what they look like when they just hatch out. They just kind of lie there for about a week until they can really swim and they've reabsorbed their yolk and they start to eat. So they're super vulnerable, not only until they hatch, but then also after they hatch. So we use the empirical relationship from my breeding experiments where I put the eggs in all those different rivers and saw how long it took them first to hatch and then to reach metamorphosis. And then I also looked at empirical scour relationships. So in all of the different monitoring that I do, I go back every week or two and say, oh, did the eggs survive? And I know if there was a spate, I know exactly sort of what ratio of discharge they were exposed to. So our metric is this ratio of the daily max to the min during the egg incubation season. And then we also assess the time to metamorphosis, again, using the data from my field experiments. And then we get three primary output performance metrics. There's the percent of clutches that avoid desiccation, the percent that avoid scour, and then how much time the little froglets have to grow in the fall, because they do a lot of growing as tadpoles, but then once they can start eating insects, they grow even faster as little froglets. And then we can put in all of these very site-specific input variables, like the rating curve, what the daily average stream flows are, and in parentheses are all the different knobs and dials, essentially, in the model that we can shift based on the local information, what's appropriate for that river. And so here's an example of the output from Alameda Creek. So on the left are what is observed, and on the right is under a proposed future flow regime, because Calaveras Dam is in the process of being rebuilt, and there are plans to restore steelhead to the stream. And right now we're trying to figure out how do you make both steelhead and yellow-legged frogs happy in the same place at the same time. So you can see that there's a much lower spike in the hydrograph, that's in blue, but it doesn't really matter because most of the eggs aren't even laid when those big pulses happen. So there was no change at all in the percent of egg masses that survive scour. That's like the green squiggly line. And there was no change in the numbers that are going to be affected by desiccation. So basically, in Alameda Creek, the biggest concern is temperature. So the red line in the top graphs is the predicted temperature. And you can see how it warms up by late summer or late spring there in June to the range that tadpoles like. Under the new flow regime, there's going to be hypolimnetic releases, and that's going to have the big effect. And it's going to take a few miles for that water to warm up, but unfortunately the frogs, their distribution in the creek kind of ends at the spot where the water warms up. So our big question is, are the frogs going to move downstream to where the water's warm? Will this just become adult frog habitat, and will they have to migrate downstream to breed, and how would they know that they need to do that? Because the temperatures aren't going to be different in the spring when they lay their eggs. The temperatures are going to be different in the summer. So we took all of these different years looking at that temperature output and combining the output of flow and temperature. What we found, could make comparisons between what were the conditions pre-dam, unimpaired, that's in black, the calculated impaired conditions, and compared to the future impaired in this new conservation strategy flow regime that's being proposed. And there were no significant differences in any of the hydrologic risk factors, but the huge differences were in temperature. And so in pre-dam conditions, it was always warm enough in 100% of the years, and now under the future flow conditions, it's only going to be warm enough 20% of the years. And as a result of this, it's directing how we have to mitigate to and make it suitable for both species. You know, what kinds of habitat enhancements could be possible to try to create some warmer areas. So that's our challenge, and the model helped us realize that. So in contrast, at the Trinity River, the big issue is desiccation. And temperature is also an issue there. But so here's an example in 2006 of how the flows declined. And just in terms of like model verification, the model did a good job of showing that any egg masses that were laid early in the breeding season, so that's the sort of that light blue turquoise line, egg masses laid on those dates are going to desiccate. And only the egg masses that are laid late in the season are going to be the ones that survive. And so part of this, using this as a tool, is you could say, well, how could we change ramping rates given the temperatures to keep these egg masses inundated long enough? And then the third place we applied this was the Tuolumne River, where right now yellow-legged frogs are super uncommon. In like 5 years of searching, we've only found tadpoles once. We've found a couple of individuals of older life stages, but they're much more common in the tributaries, like in the Clavey River and Drew Creek, and it's much warmer in the Clavey. But the Tuolumne does warm. Those are the blue lines in the thermograph here. It does get into that thermal preference range shown by the gray box. The question that we used the model to answer is, well, is there anything we can do to improve the flow regime on the Tuolumne, or could it help us explain why are they so rare on the Tuolumne? And so we could use it as a retrospective tool to say, were historic conditions more favorable? So we compared the existing conditions to the calculated unimpaired for different water year types, an extremely dry, a normal year. And what we found is that even in the unimpaired snowmelt hydrograph in the Tuolumne, it was probably not great conditions for the frogs to successfully breed, because it's a fairly steep snowmelt hydrograph, and actually in the present impaired conditions, there's a net increase of 20 more days when eggs could survive. And in a dry year, it would be 22 more days. So it could be that instead of going down a long road of like, oh, we have to rearrange a whole flow regime to try to make it better for frogs, we can say, well, it may not be that every single river, even in its unimpaired state, was appropriate. And that makes sense, right? We don't see uniform distributions of animals. And so we've found that this tool can be very useful to answer all of these different kinds of questions. We can conduct gaming of alternate flow and thermal regimes. We can evaluate the responses to change in channel geometry. So if we wanted to do some sort of a restoration and we're going to change the shape of the channel, you could say, well, what is that impact going to be? And another thing is that now that we have these different survival terms, it could be fed into other more population dynamics type models. And there are a lot of opportunities for us to expand the model, and one thing I would love to do is try to incorporate food quality issues. And I'm going to end with one really cool story from the Middle Fork American. So if you guys remember, back at the beginning of my talk, I showed you that really extreme hydrograph of daily power peaking. Well, that daily power peaking also creates big changes in the kinds of algae that grow in the river. The Middle Fork American has what's called rock snot. I don't know how many of you have heard of it. It's this algae. It's a diatom called Didymosphenia geminata, and it's really huge. If you look at the scale bar, all of these micrographs have a 10-micron scale bar in them. So you can see this is a really enormous diatom compared to the other ones, the epithemia, the superfood. And we forced tadpoles to eat Didymosphenia against their will. We put them in enclosures where they only had Didymosphenia or where they had epithemia, the really good, high-quality food, or other stock diatoms. And we reared them at two different temperatures, and that's what's in the lower right graph. And what you see plotted on the y-axis is the tadpole growth. So when the tadpoles were forced to eat this really mucousy Didymosphenia, because even though the cell is big, it lives on the end of a stalk, and they make lots and lots of gelatin, and it could be that mucus helps them as flows fluctuate. People who study diatoms have found that in response to flow fluctuation, diatoms will grow more and more stalk. And all of this mucus is really bad food. It just has no nutritional value. And as a result, tadpoles can't grow when they eat it, especially if it's cold. They lose weight, so they're below that zero line. So on epithemia, superfood, it could be cold, but they can still maintain their weight. But even if temperatures are perfect, if they eat Didymosphenia, they can't grow. And then other native stock diatoms are also bad as tadpole food. So I think this is another way of sort of bringing things full circle, of connecting the dots from the really literally microscopic scale of what diatom cells are tadpoles eating and how that scales up to their fate over their whole distribution in California. So in summary, I'm just going to review. I've covered the really big scale changes that are happening across the range of the species over a long time period, that they're absent, and the largest influence of their sites where they're absent appears to be dams. They're also missing much more prominently in Southern California, and that could be as a result of climate. And there's a grad student right now, Andrea Adams, who's looking at museum specimens that maybe chytrid fungus was responsible for their loss in Southern California. But generally, the regulated sites are colder, and there are smaller frog populations there. At the intermediate time scale of population trajectories, I went over how variable the populations can be. They do have an incredible capacity to rebound. And then at the very small scale, There are these stage-based hydrological impacts that can happen over very short time periods, but when they happen year after year, they scale up. And the survival is linked both to the hydrologic variability, the temperature, and what I think most importantly, the food. And the early life stages are the most sensitive ones. That's my story of frogs, everything I've learned about frogs in 20 years in 1 hour. So if you have any questions, I'm happy to answer them.
So if anyone in the room has questions, I'll pass the microphone around so that WebEx can hear you. Are there any WebEx questions? Okay.
So do any of you have Foothill yellow-legged frogs in the projects that you're working on? Yeah? What project?
I study trout, so. I study trout, so I do a lot of trout surveys, and
What kind of issues come up, regulatory issues come up for you when your projects you work on? Yes?
Thank you.
On Alameda Creek, I know red-legged frogs are also addressed by the conservation plan there, and I was wondering how are foothill frogs being addressed with, as you said, steelhead and red-legged frogs and other species?
Right now, I was actually just at a meeting yesterday where a bunch of we brainstormed a bunch of different and presented a bunch of different types of habitat enhancements that might be possible to try to get some heterogeneity back in the channel. In Alameda Creek in particular, because the dam actually is holding back water from Arroyo Hondo, which is draining a much bigger watershed on Mount Hamilton than Alameda Creek. So the stream is really deprived of large channel-forming flows, and the encroachment of alders in the channel has really limited the amount of habitat area. And so part of what we're thinking could potentially be a mitigation would be to try to gaps in the canopy and try to reactivate some of the bars, because the bar habitats that the frogs would normally like, and also the kinds of deep scour pools that the red-legged frogs use on the creek, are really lacking as a result of, it's just like a wall of trees on either side of the channel. So I think a lot of it is going to have to, I don't think can be, the channel morphology can't be just by flows alone. There's just not enough capacity with the dam there and the flows that can come down through Alameda Creek. And the dam does not spill very often, so I think it's going to have to be more like, physical habitat enhancement. Get some create some physical heterogeneity, scour pools and active bars, but we're trying to figure it out. It's a problem. It's conservation in action of trying to of all the different species that are there. Turtles, which are also a state special concern, are also in Alameda Creek and rely on similar habitats as the frogs.
Any other questions?
question from WebEx for you. And the question is, what kind of impact do we think chytrid fungus has on foothill yellow-legged frog populations?
Well, as I was saying, it's a very active area of research. And in Alameda Creek last fall, for the first time anywhere for boylii, we did see a die-off. And it was mostly very small individuals in December. We hadn't gotten very much rain, and all of the recently metamorphosed frogs were in literally puddles where the stream had shrunk down into little tiny pools. And I think they were just kind of sitting in zoospore soup. And so we were finding dead individuals, we swabbed them, and sure enough, they had very high levels of chytrid fungus. But interestingly, as soon as it started to rain and we sampled again in December, and then it got super cold and we weren't able to catch any frogs again until, I think, February, but almost immediately we were not picking up chytrid months of the rain starting. So I think it could be a drought stress issue. But then interestingly, Andrea Adams, a grad student from UC Santa Barbara in Sherry Briggs' lab, came up and did some sampling in August. And so we were sampling the first metamorphs and the boylii tadpoles. They were all negative, but the bullfrog tadpoles that were in the system had high positive zoospores. So it seems that the bullfrogs could be a reservoir, that boylii, it kills certain susceptible individuals, but then they're able to clear themselves of the infection, but that the bullfrogs keep it longer. And it's something we're actively trying to figure out right now. And I was fully anticipating to see numbers of frogs in the area where we had seen the die-off, but, like those population trajectories I showed you, so the area downstream where we found the chytrid, actually, relative to previous years, there were more frogs than we had seen. And so it could be that it's going to take a couple years for us to detect the effects of chytrid. So it might be that the chytrid killed off the 2013 cohort, but and it takes the females 2 years to mature. So it wouldn't be until the 2015 egg mass counts that then we would actually, if that chytrid die-off really had a long lasting. But at my population in the South Fork eel, I've found low levels of chytrid on individuals, but I've never seen a big die-off, and the population is fairly robust. So it's not a terribly satisfying answer. To be determined how it affects them.
Okay. And I have another question for you from WebEx. Are there any restoration projects you can suggest that would benefit both the salmonids and yellow-legged frogs?
Well, I think creating habitat heterogeneity is probably the most important thing. As in Arroyo Hondo, upstream of Calaveras Reservoir, there are resident trout in reservoir that swim upstream to spawn. So they're treating the reservoir like it's their ocean, upstream and spawn. And in this, in the unregulated channel where it has a very natural asymmetrical cross-section, where there's sort of a deeper pool at one end and a shallow area at the other, they were coexisting. The trout were spawning over here, the frogs were spawning over here, and they seemed okay. I think it's really about restoring natural heterogeneity, having sediment transport working in the system.
So I work in
in the North Central region, and my county is El Dorado County, and so I write a lot of CEQA comment letters. I'm just wondering, you mentioned that a female had been marked going 7 kilometers, but what is the normal sort of distribution from a water source? Do they regularly disperse between different streams, or?
Generally, they move along stream channels, but there's also a fair degree of mystery about the metamorphs. There's this one paper from 1964. I don't know if any of you are familiar with this herpetologist named Trinity. He's a lot of news, like looking at their homing. He would like blind them or do things to their sense of smell. Could they still get home? And so he put out these big arrays of pitfall traps to try to capture Kupferberg, the red-bellied newt. And he actually, in the fall, just inadvertently ended up capturing a lot of Rana boylii metamorphs. And he captured them, in these pitfall traps, up to a couple hundred meters upslope and away. Everybody always thought, boylii always hops along the stream channels. They always stay within a couple meters of water. And nobody, except for that one are the metamorphs. So there's just, I can think of a few other, actually maybe exactly in your district. So there are tunnel adits along Silver Creek that are pretty far above the right along the road. You're going to sample the reach that has frogs, and there's this big opening, and it has a little puddle of water in front of it. It's on a south-facing slope, and literally, it's a several hundred-meter talus slope between there. And in the winter, I've been there, and Ryan Peake has also been there, and found frogs and little frogs in this puddle in front of the adit. You shine a light in behind the gate in the adit, and you see a lot of light shining. So somehow, upslope movement, refuge away from high flows in the winter. Not a very good answer, but so there are these anecdotes of them moving water, but generally, I would say most individuals are moving less than a kilometer. But it's a mystery. Guys go because they have to. Sometimes they go to. You'll find them in springs or grow up there as a tadpole. They have to come. A lot of anecdotal observations far from water, but they're usually in a wet place. So how they got from where they grew up as a tadpole to that place? A little wishtail garden.
You have a question. So you mentioned that frogs or females that lay their eggs early their eggs are more likely to desiccate. So is there any evidence that behavior of laying later could become more common?
I've thought about this a lot because the other part of the pattern, I didn't go into this, is that I've also measured the sizes of the clutches as you go through the breeding. Largest females breed first. So it doesn't, so I thought about it, well, how could it be that the most fecund females that are laying the most eggs are doing the riskiest strategy? And it's got to be that there's a huge payoff in terms of priority, competition between the tadpoles. That by laying your eggs early, that there's a huge I have not seen of any change when they first start breeding from here. Could be that it really, maybe the huge and how much time have to grow before winter starts. I think that whatever the payoff is for must be huge. Otherwise, I don't understand why.
Any other questions? No? Well, thank you for the talk.